Hello everyone, welcome back to my channel Crack with Trick by Shiva. So today we are moving on to another topic of biotechnology principles and processes. Okay, so the topic what we are going to discuss today is actually amplification of gene of interest by using PCR. See, this topic is actually having high weightage. Already I told you that the chapter is actually, it is very important. Biotechnology principles and processes, biotechnology application. So approximately 11 questions are continuously every year they are asking more question from this chapter biotechnology. So this is also one of the important topic for this upcoming examination. So keep in your mind, don't ever forget it. Okay, so just keep in your mind PCR, polymerase chain reaction. Okay, so we can start from this terminology itself, amplification of gene of interest. So what do you mean by amplification? Amplification. See, last class we were discussing about actually gel electrophoresis. Okay, I hope you remember, right? Do you remember the topic, gel electrophoresis? So in gel electrophoresis, I told you that we are actually separating gene of interest. Okay, so the gene which is required for the recombinant DNA technology, that is actually we are separated by using this gel electrophoresis. Now that particular gene, we have to make it as a multiple copies. That is actually amplification. So amplification of gene of interest. Gene of interest is actually the desired gene by using. Now how we are making or how we are amplifying the DNA? By using PCR. That's it, polymerase chain reaction. Okay, now what are the major requirements? Okay, what are the major requirements of this PCR? So before going to that, you just keep in your mind Kary Mullis, the scientist who developed PCR. Okay, so Kary Mullis is the scientist who developed PCR. Now, what are the major requirements of this PCR? First requirement, DNA polymerase. So it is an enzyme. Okay, DNA polymerase enzyme. As we all know that this enzyme, it is required for making a complementary strand, right? We have already studied DNA replication in molecular basis of inheritance. So for making a complementary strand, we required an enzyme. That enzyme is actually DNA polymerase enzyme. So here also, as like the replication, the same process is actually take place. So for making a new DNA fragment, we required an enzyme. Now, one of the problem in which the Kary Mullis and his colleagues were faced was, see here, DNA polymerase, actually PCR process, it will be take place in high temperature. So if we are using an enzyme, the enzyme it get denaturated during high temperature or in high temperature. So that was the one of the problems. So continuously they need to add the DNA polymerase. Okay, so after that, see, scientists they were discovered a DNA polymerase enzyme from thermostable bacteria. For example, we can say that Thermus aquaticus. Okay, so this bacteria which is having the capability to withstand approximately 95 degrees Celsius of temperature. Okay, so here Thermus aquaticus, so the polymerase which is extracted from Thermus aquaticus is actually Taq polymerase enzyme. Okay, Taq polymerase, as like that Ven polymerase enzyme is also there. Okay, so an enzyme is required, that is actually DNA polymerase enzyme. Then a DNA template, that means a fragment of a small DNA fragment, a template DNA fragment which is required. Okay, so it is actually the fragment of DNA which has been amplified. Okay, now deoxyribonucleotides actually the substrate. So the substrate it is required for making a new strand. Okay, that is actually deoxyribonucleotides. Then a pair of primer. So a primer is actually an oligonucleotides. Okay, a primer is an oligonucleotides. So a primer with the free OH end is actually required for this reaction. So a pair of primer is also required. Now let's go for the major processes of PCR. See, this PCR, it is actually having three important steps. One is actually denaturation, then annealing, then extension. Okay, so what was the step? Denaturation, annealing, and extension. Now, what is actually the denaturation? Denaturation means just consider this as the DNA template. Okay, the DNA template, as I have already mentioned. Now, from this DNA template, we are going to amplify. Okay, now see this DNA template, it is actually treated with approximately 94 degrees Celsius. So when we are treating with the 94 degrees Celsius, what will happen? See, the bond, the hydrogen bond, it will break. Okay, so the hydrogen bond will be broken down and the double stranded DNA will become single stranded. Okay, so you can look at here, two single stranded DNAs are there. There is no connection. Hydrogen bonds are actually, it is broken down. Okay, now the second step, that is actually annealing. See, it is done slightly lower temperature than the first step, approximately 50 to 60 degrees Celsius. Okay, now in annealing process, when they are actually cooling the temperature down, at a time a primer, a pair of primer is actually added. See the green colored, that is actually the primer. As I have already told you that it should have a free 3' group. Okay, a 3' OH group is required. Now see, that is actually annealing. So the primer, it will be attaches with the single stranded DNA. Okay, now after that the last step is actually extension. In extension process, see, already I told you that deoxyribonucleotides are there and then DNA polymerase enzymes are there. So these DNA polymerase enzymes, they will add the deoxyribonucleotides on the free 3' group of the part of the primer. So when they will add the nucleotide, the primer, it get extend as like this. See here, the primer will extend in this way. So the primer will extend, they will add a nucleotide, primer will extend, and we will get two separate DNA. Have you got it? Okay, so this is actually the last step, that is actually extension. Okay, so in denaturation, the double stranded DNA is treated at a high temperature and the bonding, it will be broken down and it will be single stranded. Then in single stranded DNA, we need to add the primer. Okay, so that is actually called annealing. Then the last step, primer will be extend. Okay, so by adding deoxyribonucleotides, each of the primer will be extend and it will make new DNA strand. Okay, so this is actually the PCR, polymerase chain reaction. Okay, now see, in 30 cycles of this PCR can leads to approximately 1 billions of DNA can be generated. Or as I have already told you that amplification can be done. Okay, now here for understanding that, we have to use a trick, that is actually 2 raised to n. Okay, so n is actually the number of cycles. Okay, so 2 raised to n, that is actually 2 raised to 30 means actually we will get a 1 billion. So if they will ask questions related to this, you have to use that trick, 2 raised to n. Okay, so these are the major topic in the the major characters in the topic polymerase chain reaction. Okay, thank you.